Okay, so our first announcement is there is a new bacon quiz available online. So if you're working on those, there's one on enols and enolates that you can do. Today we're going to finish chapter 24. We'll finish our discussion of the Claisen reaction. Then we'll discuss the Michael reaction and the Robinson annulation, a lot of named reactions today. And then at the very end of class, we will start chapter 25, beginning our discussion of amines. So we left off with the simple Claisen reaction from Monday, shown here on the screen, which is a nucleophilic substitution reaction involving an ester enolate reacting with another molecule of the same ester. And then we have, of course, the acid-base chemistry that makes that an irreversible process. So, much like the aldol reaction, a crossed Claisen reaction between two different esters is only useful under certain circumstances. One of those is the same as it was with the aldol reaction. If one of our esters If only one of our esters has alpha hydrogens and the other one does not, we can perform a high-yielding and selective crossed Claisen reaction as long as we use the ester without alpha hydrogens in excess. So here's an example of that process right here. Only one enolate will form from these two esters. And as long as we have the ester without alpha hydrogens in a large excess, then that enolate is going to react preferentially with a molecule of that ester as opposed to another molecule of itself. So that will allow us, and of course we have to add acid in a second step to protonate the product. We get the enolate as the initial product of the reaction. And then our second type of cross-Claisen reaction that is useful is going to be one involving a ketone and an ester. So the standard pKa value that your book gives for a ketone is slightly more than 19. And then the standard value for the pKa of an ester is 25. Okay? So that almost 6 order of magnitude difference means that we can form a ketone enolate in the presence of an ester. And that will allow us to perform a crossed Claisen reaction. Even though it uses a ketone, it's still called a Claisen because the electrophile is going to be an ester. We'll show you how this is going to work. So here's our ketone. We'll use cyclohexanone. We'll just use a generic ester in this particular case. We're going to use our sodium ethoxide. We're going to add acid. And so what's going to happen is we're going to have a small concentration of our ketone enolate, maybe 1% or so at equilibrium. The other 99% is still going to be the ketone. And then we're going to have a really, really, really small concentration of the ester enolate because of this difference in pKa's. Okay? So the book tells you that this reaction works the best when your ester does not have alpha hydrogens like the first example. That's true, but it's not essential. You'll still get a reasonable result if you use any type of ester. So we're going to make our ketone enolate. Only one enolate is going to be in there. That enolate will then have a choice of reacting with another molecule of itself. That would be a self-aldol reaction of a ketone. Or it could do the cross-Claisen reaction and react with the ester. It's going to react with the ester, and I want you to think about why that is as I draw the product. And then once I'm done drawing the product, hopefully somebody's willing to explain why this reaction prefers to give us the crossed Claisen product as opposed to the self-aldol of the ketone. Okay? And notice that this reaction gives us a 1,3-diketone. Instead of giving us a beta-ketoester, it gives us a 1,3-diketone because we used a ketone enolate as opposed to an ester enolate. Okay? So why does this prefer the crossed Claisen as opposed to the self-aldol of the ketone? Any ideas? If not, then I'll ask you a leading question. Is the self-aldol reaction of a ketone thermodynamically favored? It is not. Self-aldol reactions of aldehydes are favored. Of ketones, they are not. What can we do in the self-aldol reaction of a ketone to drive it to completion? What irreversible process can we perform to drive a self-aldol of a ketone to completion? Yes. Dehydration, exactly. So if we want to perform a self-aldol with a ketone, we need to add heat to perform the dehydration in order to get the product. So if we do not add heat, that ketone self-aldol is going to be thermodynamically unfavored. The Claisen reaction is irreversible. We don't have to worry about the thermodynamics. Why is the Claisen reaction irreversible? It comes back to our favorite subject, the one that we struggled with on our last exam. Acid-base chemistry. So we generate this product, right? Our ketone enolate attacks our ester. We spit out ethoxide as a leaving group. We generate this product in the presence of ethoxide, and what happens? What does the ethoxide do? It deprotonates the alpha hydrogen, and that's the irreversible part because we make a resonance stabilized enolate that is less basic than the ethoxide we use to generate it. So there's two reasons why this crossed Claisen reaction occurs in preference to the self-aldol of the ketone. One is that the self-aldol of the ketone is thermodynamically unfavored unless you heat it up and perform the dehydration. The other is that the crossed Claisen reaction is irreversible because we have that acid-base step involved, okay? And so as long as there's no heat, the irreversibility of the crossed Claisen directs these two reactants down that pathway and in preference to the self-aldol of the ketone, okay? And then you might wonder, what about a self-Claisen? Well, because you would have such a teeny tiny concentration of the ester enolate, the self-Claisen is going to be suppressed, right? This almost 6 order of magnitude difference in acidity is going to give us just a negligible amount of that ester enolate when compared to the amount of ketone enolate that is present. Any questions about how this particular crossed Claisen reaction works? Okay, so there are two reactants that will participate in a different type of crossed Claisen reaction, and they're closely related. They're actually one oxidation state higher than an ester. This molecule is called ethyl chloroformate. Okay. This is at the oxidation state of CO2, carbon dioxide. It has 4 bonds between the carbonyl carbon and electronegative heteroatoms, as opposed to the 3 that we have with esters and other carboxylic acid derivatives. And then a closely related compound that actually will give the same types of products is called diethyl carbonate. Okay. So these species will react with enolates from esters or ketones in a crossed Claisen sort of process. Okay. So Let's perform a cross-clays, and let's use ethyl acetate. We're going to react that with diethyl carbonate. We're going to use the same reaction conditions that we do in a typical Claisen reaction, sodium ethoxide, followed by the use of acid. So we're going to make, we only have one enolate that we can make. We're actually going to use excess of this diethyl carbonate species. So under these conditions, we're going to make the enolate of our ester. As long as we have excess of our diethyl carbonate, that enolate will react with the diethyl carbonate as opposed to another molecule of itself. And then it's going to displace one of our ethoxy groups. But because there's two, the other ethoxy group is going to remain. So instead of giving us a beta ketoester, because our electrophile is one oxidation state higher, we're now going to generate a 1,3-diester. We're going to generate diethyl malonate in this reaction. Okay, so if you had to synthesize your diethyl malonate, this is a way that you would be able to do that. Okay, a crossed Claisen reaction between ethyl acetate and this diethyl carbonate. Okay, and of course we need our acid. Our ethoxide leaves. That ethoxide deprotonates here, giving us the enolate. And then if we don't want the enolate, if we want the 1,3-diester, we have to add the acid in order for that to work. Okay. So we can also do this with ketones. We'll draw that over here. So we'll just use cyclohexanone as our example. This time we're going to use ethyl chloroformate because we'll show you the difference. There's one difference between using ethyl chloroformate versus diethyl carbonate. We use our sodium ethoxide. And in this case, we're going to generate a beta ketoester. Okay. You'll notice I didn't write acid here. In this reaction, you don't actually need the acid. And I'll show you why. So we're going to make a small amount of the ketone enolate under these conditions. That ketone enolate is going to attack the carbonyl of our ethyl chloroformate, and it's going to give us a tetrahedral intermediate. Okay, we've got a couple choices here, but what's the best leaving group on this tetrahedral intermediate? The chloride is. Okay, so we're going to eliminate out the chloride. That's going to give us our product, which is a beta ketoester in this case. We've got our acidic alpha hydrogen. We've produced chloride. Is chloride a strong enough base to deprotonate that alpha hydrogen? I see some head shaking. It is not. Okay. So in this case, the leaving group does not act as a base, does not deprotonate the alpha hydrogen, so you don't need to add acid. You're going to get the desired product directly. Any questions? So only with ethyl chloroformate, you don't need to add the acid. In all other Claisen reactions, cross-Claisen reactions, you have to add the acid in order to protonate that enolate product. So you see that the Claisen reaction is very versatile because it allows us to form a lot of different types of 1,3-dicarbonyl compounds. The standard Claisen reaction is going to form beta ketoesters. If we use a ketone as the enolate component, we're going to get a 1,3-diketone. If we use one of these electrophiles at a higher oxidation state, that will allow us to generate a 1,3-diester. So we can make 1,3-diesters, 1,3-diketones, or beta ketoesters by choosing the appropriate nucleophile, either an ester or a ketone, and the appropriate electrophile, either an ester or one of these two compounds that's at a higher oxidation state. Any questions? All right, so the next reaction we're going to talk about is called the Dieckmann reaction. but it's simply an intramolecular Claisen reaction. I'm not sure how Dieckmann got his name attached to a reaction that was already named after somebody else, because it's the same as a Claisen reaction. It's just happening in an intramolecular sense, forming a new ring. So the mechanism you see here is the same as the Claisen mechanism, but you see that they've tried to save space here, and they've kind of drawn a cycle instead of drawing the beta ketoester twice, because the beta ketoester is both an intermediate in the reaction and then the final product. They've just kind of circled back to it. But by doing that, they've kind of obscured a key detail. They've drawn ethoxide here. So ethoxide appears as a byproduct along with the beta ketoester. That is only true for step 3. In step 3, when we eliminate out ethoxide, that's a byproduct. Okay, and then it goes on to deprotonate and give ethanol as a byproduct. What's the byproduct of step 5? Water, exactly. So by saving space and only drawing the beta ketoester once, they obscured the fact that water is the byproduct of the final step. So hopefully you understand that, but make sure you're not deceived by this attempt to kind of condense the mechanism down. All right, so a Dieckmann reaction, much like the intramolecular aldol reaction, the Dieckmann reaction works for forming 5- and 6-membered rings. Okay, so you see the example here is generating a 6-membered ring. If we had one less carbon in the tether between these two esters, we would get a 5-membered ring, and that would work. But other rings, either larger or smaller, you're going to have a strain, and those rings are going to be less stable. So we can also, the regular Claisens and the cross-Claisen reactions we can perform between a ketone and an ester. And we can do the same thing in a Dieckmann reaction. If we have a ketoester, not a beta ketoester, but a ketoester with the ketone and the ester further apart from each other in the molecule, we can perform a Dieckmann reaction. Okay, so here's an example of that. We treat this molecule with sodium ethoxide. We add our acid in a second step. So how many different enolates can we make from this ketoester? How many enolates are possible? 3 possible enolates. How many of those possible enolates could make a 5- or a 6-membered ring? Just 1, okay? So we'll have an equilibrium between those enolates. The ester enolate will be really, really, really small because of what we talked about a few moments ago. But you'll have an equilibrium between your 2 different ketone enolates, but only one of them, only the enolate at this spot, can cyclize to make a 5-membered ring. So that's what will happen. And then we know that the Claisen reaction is irreversible. We generate that enolate, and then we protonate it. So we're going to get a 1,3-diketone because we're using a ketone. Enolate is the nucleophile reacting with the ester. 5-membered ring, we count 1, 2, 3, 4, 5. So here's our 5-membered ring with our 2 ketone groups. This methyl group right here is just along for the ride. It's going to be outside the ring. It's not going to be part of the 5 carbons that form the ring. So that methyl group is going to go right here because this carbon is the carbon that becomes the alpha carbon in between the 2 carbonyls. And so that carbon is going to bear the methyl group in the product. Sometimes when you're drawing products from these reactions, particularly cyclization, sometimes it's hard to figure out exactly where the carbons are going to go. In those cases, it's helpful to number the carbons in the starting material. If you number the carbons in the starting material, you see which carbons are bonded to which other carbons, and then that would help you recognize that this methyl group needs to be attached to that carbon in the product. Okay? Are there any questions about the Dieckmann reaction? Okay, so aldol reactions and Claisen reactions are very common in biochemistry, and we'll look at a few examples of those here. So there are some differences between aldol reactions in biochemistry versus aldol reactions in a lab or in a flask. One is that in biochemistry, most organic reactions are performed in a fairly narrow pH range. And so we're not going to have super strong bases available to make enolates. There's not going to be any LDA in nature. We're not going to have ethoxide. We're going to have very small, very low concentrations of hydroxide at the pHs that these reactions operate. So we use enols as our nucleophiles as opposed to enolates. Another thing we've learned about biochemical organic reactions is that we can have more than two components reacting simultaneously. We don't do that in a flask, but in the confines of the active site of an enzyme, the non-covalent interactions such as hydrogen bonds that those enzymes can make to the reacting molecules can hold them in close enough proximity that we can often have more than two species coming together in a single step. So we see that here in this biological aldol reaction. We're using dihydroxyacetone phosphate. We're making the enol in one step. To go from ketone to enol, we have to deprotonate the alpha hydrogen, protonate the carbonyl carbon. We can actually do those simultaneously because the enzyme active site positions a base in close proximity to the alpha hydrogen and an acid in close proximity to the carbonyl oxygen. So this works, but only in within the confines of the active site of the enzyme. And then once you get this enol, we're calling it an enediol because there's two OH groups involved due to the presence of this OH group in the reacting ketone. Then that is going to add to the aldehyde of glyceraldehyde 3-phosphate, which is also held in the active sites in close proximity to that enediol. There's going to be a base involved, but we don't have an enolate intermediate because the base deprotonates the enol simultaneous to the enol attacking the aldehyde. And we don't get an alkoxide because we're protonating that oxygen simultaneously. So things that would take 2 steps or 3 steps in a reaction vessel in a flask are going to take 1 step within the confines of the active site of an enzyme. Another interesting feature here, we mentioned previously to you that aldol reactions frequently form multiple stereocenters. And this reaction is no exception. We have a chiral aldehyde, so this stereocenter remains in the product. But these two new stereocenters are new. They're created by virtue of formation of that carbon-carbon bond. And you notice that only one isomer is formed. Only one of the 4 possible diastereomers that you could generate from this reaction is formed. And that's because the active site of the enzyme is going to hold those reactants together in a conformation that will only produce the desired isomer. Okay? So this reaction is making fructose 1,6-bisphosphate, which the body can then convert into glucose. So this is a key step in gluconeogenesis, or the biosynthesis of glucose. So aldol reactions, very commonly used in biochemistry to form carbon-carbon bonds. Here we're linking 2 3-carbon units together to make a 6-carbon species that can be turned into a 6-carbon sugar, which in this case is glucose. Any questions about this biological aldol reaction? Okay, here's another one for you. This is the formation of citrate, conjugate base of citric acid, which is a triacid. And here we're making an enol from a thioester, acetyl coenzyme A, which is a very common 2-carbon building block in nature. And we make that enol in the same fashion as we saw on the previous slide. We perform the aldol in the same fashion, where we have the proton transfer steps occurring simultaneous to the carbon-carbon bond forming steps. And that gives us citryl coenzyme A, and then that thioester can be hydrolyzed to generate the citrate. Okay. So sometimes in nature we use aldol reactions to form carbon-carbon bonds, but we learned at the beginning of the chapter that the aldol is a reversible process. It's a thermodynamic reaction, reversible, and so retroaldol reactions are also common in biochemistry. Sometimes when we are metabolizing molecules, when we're trying to break them down into smaller subunits, we need to break carbon-carbon bonds. And one of the best ways to break a carbon-carbon bond is via a retroaldol reaction. So this retroaldol reaction that we see here is simply the reverse of the aldol reaction I showed you 2 slides earlier. We have fructose 1,6-bisphosphate, which is derived from glucose in the glycolysis pathway, something we'll study later on this semester. Glucose gets converted into fructose 1,6-bisphosphate. The body needs to chop that into 2, 3 carbon species in order to get energy from it, and it does so via this retroaldol reaction. So the arrows are simply going in the opposite direction as they did in the aldol. We're deprotonating this oxygen, but we're not generating an alkoxide. We are eliminating out an enolate, but it's not an enolate. We're protonating the oxygen, so we end up eliminating out an enol. So these arrows are just the reverse of the arrows we saw when we coupled these 2 molecules in the aldol to give this product. Okay? So it's very common in nature for enzymes to catalyze both a forward reaction and a reverse reaction of an equilibrium process. And the direction the reaction goes is going to be dictated by the concentrations of those components in the cell. So if your body has a lot of glucose, then the enzymes are going to break down that glucose and generate energy from the glucose. And you're going to have glycolysis, and you're going to see this reaction. Conversely, if your body is low on glucose and needs glucose, then you're going to have gluconeogenesis. You're going to be doing the aldol reaction and generating the 6 carbon species. Okay? Any questions? All right, then we'll also look at a biological Claisen reaction. Claisen reactions are used in nature to form 6-membered rings, or not 6-membered rings, that would be an intramolecular Claisen, to form carbon-carbon bonds, is what I meant to say. And this actually shows you an interesting way of making an enolate, the way that enolates are made in nature. I told you that we usually see enols as opposed to enolates as our nucleophiles in biochemical reactions. That's because we don't have LDA available, and we have usually very, very small concentrations of hydroxide available. So it's not practical to make enolates under physiological conditions, unless you form an enolate via decarboxylation. Okay? So we know that 1,3-dicarbonyl compounds, if it's a carboxylic acid or a carboxylate in this case, can undergo decarboxylation. That requires heat beyond the temperatures that are available in living organisms. However, this can be catalyzed by enzymes. So within the confines of an enzyme active site, that enzyme active site can lower the transition state energy, lower the activation energy for decarboxylation, and allow decarboxylation to occur under physiological temperatures, generating an enolate. So whenever we use enolates in biochemistry, they're almost always obtained from a decarboxylation, as we see here. So this is giving us a thioester enolate that is able to react with another molecule of a thioester. So the Claisens we do in the lab usually involve esters. Nature tends to use thioesters more than esters, and so biological Claisen reactions frequently involve 2 thioesters. And so here that's generating a beta-keto thioester. And that's a key process in the biosynthesis of fatty acids. When we have our lipids chapter towards the end of the semester, we will discuss this process in more detail, and we'll show how this beta-keto thioester gets turned into a thioester itself, how we get rid of this carbonyl. Okay? It takes 3 steps to do so. Any questions about our biological aldol or Claisen reactions? Okay, so let's talk now about a reaction called the Michael reaction. So The Michael reaction is one of the few reactions named after an American. Arthur Michael was a chemist in the late 1800s and early 1900s. He worked at Harvard for a time. And Arthur Michael lived the dream of many men in that he married a wealthy woman. And so what would you do, guys, if you married a wealthy woman? Quit your job, perhaps? Do whatever you want? Oh, that's what he did. But what he wanted to do was organic chemistry research. So he just set up his own organic chemistry lab and performed research. And it's actually romantic because his wife was also a chemist. And so they worked side by side in the lab, just doing research for fun because she had enough money to support them in that way. So while he was doing this, he discovered a reaction that now bears his name called the Michael reaction. And the Michael reaction involves alpha-beta unsaturated carbonyl compounds. So let's review something we learned earlier about alpha-beta unsaturated aldehydes or ketones. We'll do a ketone. These don't have to be methyl groups. They could be any sort of substituents coming off of that double bond. So we know anytime we have a carbonyl, we have this minor resonance contributor that we can draw with the negative charge on the oxygen and the positive charge on what was the carbonyl carbon. But when we have an alpha-beta unsaturated compound where we have conjugation, that gives us an additional minor contributor, which we can draw like this. And this minor contributor has the positive charge on the beta carbon. So when we draw the hybrid structure, we have a lot of dashed lines like this. We have a partial negative charge on the oxygen, partial positive charges on the carbonyl carbon and the beta carbon. So we use these resonance structures and the hybrid structure to indicate that our alpha-beta unsaturated carbonyl compounds are electrophilic at two spots. Okay? And we learned earlier on, I think it was chapter 21, where we learned that if we use organocuprates, those organocuprates will add to the beta carbon of the carbonyl, whereas Grignard reagents and organolithiums add to the alpha carbon. So enolates are another type of nucleophile, and Michael learned and discovered that enolates will add to the beta carbon of these alpha-beta unsaturated compounds. So we can take, this is our simplest alpha-beta unsaturated ketone. It's called methyl vinyl ketone. We can react that with a compound that can make enolates. Usually we either use a ketone or we use a 1,3-dicarbonyl compound. The 1,3-dicarbonyl compounds are more common, so that's what we'll use in the examples we'll show you. We use our sodium ethoxide, and then we add our acid, and that is going to form a carbon-carbon bond between the alpha carbon of our 1,3-dicarbonyl compound and the beta carbon of our alpha-beta unsaturated ketone. So the product will look like this. Okay. So here's our carbon-carbon bond that we formed. This was the alpha carbon of our 1,3-diester. This was the beta carbon of our alpha-beta unsaturated ketone. So what's happening in this reaction? Let's go ahead and draw the mechanism. Let's raise this up just a little bit. So we're going to start with our 1,3-dicarbonyl compound. Okay. I need to give myself more room. So we've seen this before. Hopefully you can predict that our ethoxide is going to deprotonate our 1,3-dicarbonyl compound and make an enolate. That's going to be irreversible because it is the resonance stabilized by an extra major contributor, and it's conjugated. Okay, so we're going to get this. We also produced ethanol in this reaction. That will come into play later on. Okay, so now our nucleophilic enolate can react with our electrophilic ketone. It could add to the carbonyl, or it could add to the beta carbon. For reasons we won't get into, it prefers to add to the beta carbon. Okay. So enolate attacks beta carbon. Pi electrons from the alkene go towards the carbonyl. And we have to push the electrons from the carbonyl pi bond onto the oxygen. This is going to be reversible. We'll use our reversible arrows to show this step. So that's going to give us, we'll turn this around. It'll be easier to draw if I turn this around. So here's our ketone. Here's our beta carbon from the ketone. And our enolate that we draw is going to look like this. And then okay. All right, so here's our intermediate that we generate where we formed our carbon-carbon bond. We formed an enolate. Remember, we generated ethanol in our first step. How are those going to react with each other? How's the ethanol going to react with our, it's going to protonate the carbon of our enolate. So let's put the ethanol over here. We'll go this way now as we draw the product. And okay, so here's the product. This is the final product of the reaction. By using ethanol to protonate our enolate, we've regenerated ethoxide. So are we finished? No, our ethoxide, what's our ethoxide going to do? It's going to deprotonate the alpha hydrogen here. Okay. So we're running out of space. So I'm just going to write deprotonation. So that's going to give us our resonance stabilized enolate. And then what do we have to add in order to get the product? What do we add as the second step in our Claisen reactions? We add acid. So we'll do the same thing here in the Michael reaction. We have to add acid at this point. And that acid is going to protonate that enolate. So we didn't draw that process out on the board here, but we've drawn it in the Claisen reaction. So you should be able to draw both of these steps. And then we're going to obtain the desired product at that point. Okay? Any questions about the Michael reaction? Yes? In the second to last step, so here, carbon, could you protonate the oxygen? You could, and that will happen, but then that'll just tautomerize to give you the ketone. So you'd get to the same place. It's just quicker to get there drawing it this way. So you could draw it the other way, but then you'd have to tautomerize that enol. Okay? So here's an example of a Michael reaction being used to form this particular carbon-carbon bond in this intermediate that was turned into estrone. All right, the last reaction we're going to talk about today is called the Robinson annulation. The Robinson annulation is named after Sir Robert Robinson. He is a British chemist. He is beloved by organic chemistry students worldwide because he is credited with inventing curved arrows. So he's responsible for you having to draw curved arrows to show reaction mechanisms. But before you complain about that, think about how difficult it would be to understand organic reactions without curved arrows, right? Even though they might not be fun to draw when you're taking an exam, they really, really help us understand organic reactions. So for that discovery, he was given the Nobel Prize in 1947. And this reaction we're going to learn about now is called the Robinson annulation. Annulation means ring-forming process. So in a Robinson annulation, we're going to generate a new ring. Like the Michael reaction, we're going to use alpha-beta unsaturated carbonyl compounds. Also like the Michael reaction, we're going to use either ketones or 1,3-dicarbonyl compounds as our nucleophiles, as our enolate precursors. Unlike the Michael reaction, we're going to use hydroxide, water, and heat. And that's going to enable us to form a new 6-membered ring. Okay? So the product of this particular Robinson annulation would look like this. The 6-membered ring on the right came from the 6-membered ring in the 1,3-diketone, and this 6-membered ring is the one that was formed in the Robinson annulation. So the Robinson annulation generates cyclohexeneones, where we have a 6-membered ring with a double bond and a carbonyl. So the Robinson annulation, the good news about the mechanism is we already know it. It's a combination of 3 processes that we've already learned. First one, Michael addition. Okay? Make the enolate, add to the beta carbon, protonate that enolate, we end up with a 1,3-dicarbonyl species. Okay, we just went over those steps. Okay, but in this case, after the Michael addition occurs, we now have an intramolecular aldol reaction. Okay, where we make a new enolate that can form a 6-membered ring, protonate to get a beta hydroxy carbonyl compound. Now this is a ketone aldol reaction. Those ketone aldol reactions are thermodynamically unfavored, so we have to add heat to dehydrate to make them favorable. So that's what happens here. We add heat, we deprotonate, make the enolate, eliminate out the hydroxide, and here we have our new 6-membered ring. Okay, so it's a long mechanism, but it's 3 reactions we've already learned. Michael, intramolecular aldol, dehydration. You should be able to draw all 3 of those. So you might ask, how would we get the process to stop at the Michael if we don't want the Robinson annulation? The answer is don't add heat. It's the heat that drives the reaction through to the Robinson annulation. So this sample problem here shows you how you would draw the products from a Robinson annulation, joining the alpha and beta carbons of the 2 reactants, and then the other alpha carbon of the ketone with that ketone to form our 6-membered ring. And then this iClicker question shows us how we would use it in a retrosynthetic fashion, going from, or looking at a product and deciding which building blocks would make that product. So let's go ahead and do this iClicker question. And we don't have a ton of time, so we do want to finish this iClicker question off, hopefully in the next 20 seconds or so, 20 or 30 seconds. Okay. All right, we'll give you about 10 more seconds. Any more answers coming in? Any more than 26? All right, that looks like that's it. Okay, 100%. Everybody went with A. Very good. So you know how to use the Robinson annulation in a retrosynthesis. Let's take about 2 minutes to introduce amines to you. Let's look at an electrostatic potential map of amines. Whoops, I didn't mean to do that. Sorry. And what we see is that they're basic. They're electron rich on nitrogen. Nitrogen is the least electronegative atom that has a lone pair. So that lone pair is more available than it is in other organic compounds. So amines are going to be more basic than other neutral organic compounds. More nucleophilic. They're going to undergo a lot of reactions that we'll cover in this chapter. This is how we classify amines based on the number of alkyl groups attached to the nitrogen. So it's different from alcohols. Alcohols, it's the number of alkyl groups attached to the carbon bearing the oxygen. Amines, it's the number of alkyl groups on the nitrogen. If we have different alkyl groups on a secondary and a tertiary amine, because the lone pair counts as a group, we might end up with a stereocenter. We could have 4 different groups on a nitrogen. But when one of those groups is a lone pair, it can switch positions rapidly. Because it switches positions rapidly, it is not a stereocenter. The only stereocenters we find on nitrogen are ammonium ions. So if we protonate an amine, and we make an ammonium salt of that amine, now we no longer have a lone pair on the nitrogen, and this can be a stereocenter. We'll stop there, continue with amines on Friday.